Welcome to Week 8 of BIOT 643. This week, we'll be discussing Part 2 of Protein Technologies. As an outline for this lecture, we'll be discussing the expression of proteins in prokaryotic systems, the expression of proteins in eukaryotic systems, including fungal, insect, and mammalian cells. We'll then discuss high-throughput proteomics. Within those, we'll be discussing the methods of mass spectrometry and protein arrays. And it's important to note that this week is Lecture 8. Because the midterm exam was held during Week 7, there is no Lecture 7. First, the expression of proteins. The goal of gene expression systems is not only to clone a gene, but also to express it. That is, to produce a functional protein. High levels of a protein production can be obtained by modifying any of the various steps, including transcription and translation. In addition, factors that promote protein stability and effective secretion of the product of the cloned gene positively influence protein levels. The desired proteins can be expressed in eukaryotics or in prokaryotic cells. Once a gene has been cloned, it needs to be expressed in order to produce the corresponding functional protein. Some of the systems that can be used for recombinant proteins are prokaryotes, such as bacteria, simple eukaryotes, fungi, and yeast, or the higher eukaryotic cells, insects, plants, and mammalian cells. Let's start with prokaryotic expression systems of proteins. To express a protein, the first organism of choice is typically a bacterium, based on the short life cycle or doubling time. It can be around 30 minutes. It's economical. It's easily scalable to generate many liters of bacterial culture fluids. And it can be engineered secretion of the protein into the surrounding medium. The first commercial success of recombinant DNA technology was the production of human insulin in bacteria in 1982. Simply cloning a gene into a vector does not necessarily mean it will be expressed. So how is a human protein produced in bacterial cells, and what are the steps involved? First, you create a recombinant molecule by cloning the gene, the cDNA or genomic, encoding the target protein into the appropriate cloning vector. Then there is transformation and screening. And third, the isolation and purification of the produced protein. So what are the advantages of the prokaryotic system? It's inexpensive, easy to manipulate, and scalable. The specific advantages of the E. coli system include a low cost, continuous fermentation, and a short life cycle. PET and other commercial expression vectors that work in the E. coli system are commonly used for protein expression. The specific advantages of the Bacillus subtilis, the soil bacterium, is that it is easily transformed, grows to high density, is cost effective, and secretes its proteins directly into the media. But there are limitations to this prokaryotic system. They lack the post-translational modifications, such that any mammalian protein that requires a post-translational modification, whether it's glycosylation, phosphorylation, or methylation for its function, may not be successfully expressed in the bacteria. They can be unstable. A number of proteins being produced in bacteria are rapidly degraded by proteases. This is particularly true when using strong promoters. There are also issues with protein insolubility. In many cases, the protein expressed in a bacterial system is insoluble, which makes it difficult to purify. The latter two problems can be reduced or eliminated by creating fusion proteins, where the protein of interest is tagged with a short sequence that makes it more stable and also easy to purify, and in some cases increases the solubility. For example, the GST fusion proteins can be purified using a glutathione affinity column. So now let's talk about the eukaryotic expression of proteins. The features of the eukaryotic system are that the eukaryotic expression vectors are essentially the same as their prokaryotic counterparts. A eukaryotic promoter sequence, a selectable eukaryotic marker gene, and eukaryotic transcriptional and translational stop signals. The sequence of events is also similar to that of the prokaryotic system. First, a vector with a gene of interest is constructed. Next, the recombinant vector is introduced into one of the eukaryotic expression systems for synthesis of the desired protein. Besides prokaryotes, other protein expression systems are yeast, insect cells, and mammalian cells. The sequence of events is similar to that of the prokaryotic system. Again, a vector with the gene of interest is constructed. Then, the recombinant vector is introduced in one of the eukaryotic expression systems for synthesis of the protein of interest. So why is this eukaryotic expression system necessary? Well, protein folding and processing is natural, such that the recombinant protein is close to that of the native protein. And even if other proteins are co-purified, there are less chances of an immunogenic response. So the three eukaryotic expression systems that we're going to talk about are the fungal expression system, the insect cell expression system, and the mammalian cell expression system. First, the fungal system. The most commonly used fungal expression system is a yeast called Saccharomyces cerevisiae. There are three kinds of expression vectors. There is the yeast expression plasmid, there is the yeast integrative plasmid, and the yeast artificial chromosome. The last two of these are part of the yeast chromosome and hence are more stable. This yeast artificial chromosome can accept large size inserts. Recombinant vectors can be introduced into yeast cells by electroporation, protoplast fusion, or lithium acetate method. The advantages of the fungal expression system are that it's single-celled and hence simple. It's well characterized genetically and physiologically. There are several strong promoters, so high levels of protein expression can be obtained. This expression can be carried out many of the post-translational modifications required to express functional eukaryotic proteins. Normally secretes very few of its own proteins, so proteins designed to be secreted can be easily purified. And they can be grown in large cultures, so scaling up is very easy. And this system is stable, it's durable, and it's cost effective. And the FDA has approved these as safe organisms. So this table lists some of the recombinant proteins that have been produced in the yeast expression systems. There are vaccines, such as the hepatitis B virus surface antigen, the malaria protein, as well as the HIV-1 envelope protein. It can be used to develop proteins for diagnostics, such as the hepatitis C virus protein and HIV antigens. They've also been used for human therapeutic agents, including epidermal growth factor, insulin, insulin-like growth factor, platelet-derived growth factor, proinsulin, fibroblast growth factor, just to name a few. Let's next talk about insect expression systems. For the insect cell expression systems, the higher order of eukaryotes than yeast, and these can carry out more complex post-translational modifications. These are used as expression systems based on the properties of baculoviruses, so specifically infected invertebrates. These systems are used indirectly in the sense that first, the viral genome is modified to create a recombinant molecule. The modified baculovirus are infected into the insect cells, which are then the host for the multiplication of the virus particles. The baculoviruses are double-stranded DNA viruses that can infect and kill a large number of insect pests. However, the cloning of certain genes into the virus can make it a useful gene delivery vehicle. Non-essential genes of the virus are removed and replaced with the gene of interest. The baculoviruses exclusively infect invertebrates, including many insects such as the moth, fruit fly, etc. On completion of the life cycle, the viruses kill the host cells and release large amounts of protein. So the steps in developing the insect cell expression system. First, the insect cells are transfected with the baculovirus. Specifically designed transfer vectors or plasmids with a gene of interest is transfected or introduced into the insect cells carrying a baculovirus. The gene of interest is transferred into the baculovirus inside the insect cell. The recombinant baculovirus, i.e. carrying the gene of interest, is identified by PCR or hybridization. The recombinant baculovirus is isolated. The insect cells are now infected with this recombinant baculovirus. Protein expression from the gene of interest is then purified. Here we show an example with initially the baculovirus DNA is cleaved. Here we see is cleaved at the site of gene 603 and gene 1629. Then you have a ligation of a transfer of your gene of interest, which is surrounded again by the gene 603 and the gene 1629 in a transfer vector. This now allows the formation of your recombinant baculovirus, which contains your gene of interest sandwiched between a complete gene of 603 and 1629. The advantages of this insect cell expression system are that it gives you the proper post-translational modifications and folding of the heterologous proteins. It has a high level of protein production, and the proteins can be secreted or targeted to specific cellular locations. The disadvantages are that In some instances, improper folding and aggregation occurs, and it comes at a relatively high cost. There are more than 500 proteins that have been expressed using this system, of which more than 95% had the correct post-translational modifications. And just to show a few, there's alpha interferon, the anthrax antigen, beta amyloid precursor protein, beta interferon, G protein coupled receptors, human alkaline phosphatase, human DNA polymerase alpha, malaria proteins, multi-drug transport proteins, poliovirus proteins, and tissue plasminogen activator. Now let's talk about mammalian systems of expression of proteins. The first mammalian cells used for the production of proteins was the Chinese hamster ovary cells, or CHO, cell lines. These systems are the ones of choice to produce mammalian proteins, but for the cost associated with them. There are several ways to transfer DNA into these mammalian cells. There's 1. The direct physical introduction. 2. Disruption of the cell membranes to allow transfer of the DNA. 3. The use of viruses as delivery vehicles. And 4. The formation of DNA complexes with inorganic salts, prokaryotics, and lipids. So mammalian expression systems utilize specially generated cell lines that are used for production of heterologous proteins. The first such cell line was the Chinese hamster ovary cell. The two-vector expression system consisted of 2 origins of replication for the 2 vectors which express a dimeric protein. The eukaryotic promoters, labeled as P, there's the polyadenylation and termination sequences, the PA and the 2 double T's, as well as selectable marker genes. So what are the characteristics of these different expression systems? If you look at cell growth, for both E. coli and yeast, these are very rapid, as opposed to insect cells, which can be slow in the range of 18 to 24 hours, and mammalian cells, which can be 24 hours or greater. The complexity of the growth media. For both, again, E. coli and yeast, it is very minimal. In the context of insect cells and mammalian cells, the complexity of the growth media is very complex. The cost of the growth media for E. coli and yeast cells, it is low, whereas for insect and mammalian cells, the costs can be very high. The expression levels in E. coli cells can be high. In yeast cells, depending upon the promoters and vectors used, it can be anywhere from low to high. And the same is said for insect cells. These can be from low to high expression levels. However, in mammalian cells, these tend to be low to moderate. And for the extracellular expression, in E. coli, the proteins are secreted into the periplasm. In the yeast cells, the proteins are secreted directly into the media. And in insect cells and mammalian cells, again, these proteins are secreted directly into the media. And in terms of post-translational folding modifications, in E. coli, these require refolding, as do the yeast system. However, in both the insect cells and mammalian cells, these both will result in appropriate folding of the proteins. So now let's shift gears and we're going to talk about high throughput proteomics. High throughput proteomics are used to identify protein molecules in complex mixtures. They can also be used to quantitate these proteins. You can identify the proteins that they interact with, either localization, modifications, and activities. There are widespread applications in diagnostics, drug discovery, and disease identification. So understanding of the protein function at the cellular level is a key to improving human life by developing medicines and identifying targets to combat new and prevalent diseases. So some of the tools for high throughput proteomics are one-dimensional and two-dimensional electrophoresis for separation and visualization of proteins. Mass spectrometry. This plays an important role in protein complex identification as well as simple protein identification. Protein-protein interaction studies. These are key to understanding the roles of proteins in normal function of a cell. Such methods that can be used are the yeast 2 hybrid method as well as the fluorescent resonant energy transfer system. Protein arrays can be used to study protein expression and protein localization. First, let's talk about mass spectrometry. Mass spectrometry is an analytical technique used for protein identification and structure identification by measuring the mass-charge ratio of ions. It is used to find the composition of a sample by generating a mass spectrum of sample components. It involves 3 steps: ionizing the sample, separating ions from different masses, and measuring the intensities of the ion flux. The instrument used for this purpose is a mass spectrometer, and it consists of 3 components: an ionizer, a mass analyzer, and a detector. This approach consists of 2 steps. Protein molecules in liquid form are converted into ions in the gaseous phase. These ions are separated based on the mass-charge ratio by an analyzer and then pass through a detector to estimate this ratio. This is diagrammatically shown on the figure at the bottom of this page. These ions can be generated by 2 methods: what is termed MALDI or matrix-assisted laser desorption ionization. In this approach, the analyte or the protein mixture of interest is co-crystallized with a light-absorbing compound. That is what we term the matrix. This mixture is placed on a stage and exposed to a laser beam of a specific wavelength, which causes the conversion of the liquid into gaseous ions. Here we show an example of the MALDI ionization. We have a laser at 337 nanometers shines on your sample, and remember, your sample is a mixture of a light-absorbing compound or matrix co-crystallized with your protein mixture. This takes that liquid form and ionizes it into gaseous ions, which then is transferred into the actual mass spectrometer. Here we show the series of steps in the events of the MALDI time-of-flight technology. And first step is your matrix assisted. Basically, your molecule of interest is co-crystallized or dried in combination with your light absorbing compound or matrix. Then it undergoes the step termed the laser desorption or ionization. In this case, a pulse of laser light is used to force the molecules into a gas phase and ionize them. This is followed by the time of flight. The ions are then accelerated using an electric field and allowed to drift down to the end of the flight tube where they strike a detector. This drift time, or time of flight, is measured by sophisticated electronics, and this is usually in the range of nanoseconds per slice. And finally, you obtain a result is a mass spectrum with a peak list documenting the masses of all of the analytes. So the generation of ions. So you have the electrospray ionization. This ESI creates ions by application of a voltage potential to a flowing liquid containing your analyte or protein through a small, highly charged metal capillary, causing the liquid to ionize and spray. In the second step, the ions are separated based on the mass to size ratio by an analyzer and then passed through a detector to estimate this ratio. The smaller the mass of the ion, the faster it will reach the detector. This is also known as mass spectrometry or tandem mass spectrometry. In this system, a sample is passed through two successive mass spectrometers. In the first analyzer, only peptides of specific mass charge ratios are allowed to pass through. In the second analyzer, the specific peptides are broken down further by collision with a gas in a process called collision-induced dissociation. Here, and we show the example where the sample is put through an electron spray ionization and MALDI, where your sample is ionized. We now have charged peptides and of specific mass to charge ratios. This within the first mass spectrometer, these peptides are separated out and then transferred to the second mass spectrometer. Now these individual fragments are then ionized. As they're ionized, this peptide is dissociated or broken one amino acid at a time. So if you have a peptide which has 100 amino acids, the first peptide that will be seen will contain all 100. As it is broken down, your next peptide will have 99 amino acids. The next peptide will have 98 amino acids, 97, and so on. And the molecular weight of each amino acid allows you to determine the sequence of each one of the amino acids within that peptide, and that allows you to define the amino acid sequence or the primary sequence of that peptide. Within tandem mass spectrometry, this method is becoming increasingly popular for its higher automation and efficiency. Using the electron spray ionization and MALDI time of flight, high molecular weight proteins, that is, greater than 100 kilodaltons, can be identified. A limitation of mass spectrometry is that many biologic proteins exist as complexes. Thus, it is possible and likely that abundant ions will be mass the fewer ones, or the spectrum is hard to interpret. Therefore, it is important to fractionate the proteins before mass spectrometry. And the two most common ways of doing that is through 1. The two-dimensional electrophoresis for intact proteins, or high-pressure liquid chromatography, or HPLC, for peptide fragments. So next, we're going to talk about protein arrays. Protein arrays used for protein expression profiling and for studying protein-protein interactions on a large scale. This approach was developed to overcome the limitations of two-dimensional gel electrophoresis, primarily the tedious analysis and difficulty to identify low abundance proteins. In principle, these protein arrays are analogous to DNA arrays, except that instead of oligonucleotides or complementary DNAs, a large number of proteins or antibodies are spotted onto the slide and then used to screen for either differentially expressed proteins, protein-protein interactions, protein-substrate interactions, or protein-ligand interactions. Here, we show an example of a protein array. In this case, this is an array for the protein receptor microarray where you have two estrogen receptors, one with a green fluorophore and the other with a red fluorophore, that are attached to an aldehyde microarray slide. The slides or receptors are exposed to different compounds to determine whether the compounds aid or inhibit the binding of the estrogen receptor to the slide. Protein arrays like the DNA arrays in that the protein chips consist of two layers. The first layer is the substrate layer, which in turn is composed of 2 to 3 sublayers. And these can have a bottom layer of glass, silicon, or synthetic polymers, which are coated with a layer of another metal, and the two can be held together with a third metal layer. The second layer of the microarray chip is the protein layer, which can consist of a tag, an adapter, and a protein. And here on this diagram we show an example of this setup. Unlike DNA arrays, these protein chips are technically much more difficult to create. And this is largely because of the tedious methods used to purify and isolate functionally active proteins and the complex nature of these proteins. However, there are many potential problems associated with this technology. First, obtaining a large array of proteins. There are difficulties with the proteins coupling to surfaces. There's dehydration of these proteins, miniaturization of the protein volumes, the availability of specific antibodies, or the cross-reactivities of these antibodies, the sensitivities and dynamic range of detection systems, and last, the standardization and data storage. There are three types of protein microarrays. There are capture arrays, where the proteins or antibodies are spotted on a solid surface and screened with a labeled mixed sample, typically termed protein profiling. There are reverse phase arrays. This is where the protein lysates, or a mixture of proteins, are spotted on the solid surface and screened with a single target molecule. And then lastly, there are functional protein arrays. Here we have open reading frames, or ORFs, are cloned on expression vectors, the corresponding proteins expressed and spotted on the slide. These are then screened as in capture arrays, but typically used for studying protein-protein or protein small molecule interactions. Here, in the first one, we show the pure proteins are arrayed on a functional array. In this case, this is a kinase array. The protein interactions are screened. You can accessing binders. And this has been typically used in diagnostics for detecting autoantibodies. The next, we show the protein binders are arranged and a capture array. And these are typically used to measure levels of proteins, the proteome expression profiling. And again, these have been commonly used in the development of diagnostics to detect biomarkers as well as to quantify cytokines. And the last example here are the complex sample arrayed on a reverse array or a lysate array. Here, this is commonly used to measure levels of protein or to identify the proteome expression profiling. And again, these have been commonly used in diagnostics to detect biomarkers. Here, we give you an example of the layout of these two types of arrays, the applications of protein and antibody arrays. For using your uniclone set or your protein expression, we can have, going down on this chart, the generation of specific binders or antibodies by library display or hybridoma technology. And then we use these to develop antibody arrays. And this can be done to assess translation profiling or the profiling of proteins present in your samples. Or we can develop protein expressions where we have the protein arrays. These are commonly used for profiling antibody binding for specificity and cross-reactivity, for profiling sera and other bodily fluids for the potential to discover diagnostic and prognostic markers, for the potential application in protein-protein interactions, as well as organ and disease-specific protein arrays. And for your review, the references that were used in the development of this lecture and other topics are as follows.